So the last part of the chapter, it includes the enzymes. Enzymes are the catalyst which speed up the rate of chemical reaction without being changed or used up in the chemical reaction. They are proteinaceous in nature, as I told you before, and these are called as biological catalyst because they are working in the biological systems. These enzymes are very necessary for the living organisms because they only speed up the chemical reactions. Otherwise, it would take weeks to digest a single meal which we eat in a day. Then, how the naming of enzymes is done? It is done according to the reaction which they are catalyzing. For example, the enzyme which catalyzes or which break down carbohydrates, it is called as carbohydrases. Which break down proteins is called proteases, and which break down lipids, it is called as lipases. And certain specific names can also be given to the enzymes. For example, if an enzyme is catalyzing a reaction of a starch, it is called as amylase. Then how do enzymes work? So the enzymes, they have a specific substrate, okay, and they can only combine to a specific substrate, and when they combine to that substrate, a reaction takes place and the substrate, it changes into a product, and the enzyme, it remains as it is. It cannot bind to all the substrates. It has a specificity. For example, in the diagram you can see that enzyme, yellow portion, that is the enzyme, it can only bind to the complementary site. It can only bind to a complementary substrate because it works like a lock and key hypothesis. All the locks, they cannot be opened by a single key, and a single key, it cannot open all the locks. So there is one specific lock and one specific key. In the same way, the enzymes also work by lock and key hypothesis. Then properties of enzymes, they include that they are proteins. They are made inactive by high temperature. They work best at a particular temperature that is called as optimum temperature. They work best at a particular pH that is called as optimum pH, and they are catalyst and they are very specific. Then what happens when on the activity of enzyme with the temperature? So if the temperature is very less, enzymes, they will not be having that much kinetic energy, so they will not collide with the substrate, and so the activity or the productivity of the enzyme will be less. Then as the temperature increases, the productivity of the enzyme, it increases, and there comes a certain temperature at which it is having the maximum capacity to catalyze a reaction. So that is known as a optimum temperature. But if that temperature is exceeded, what happens? The proteins, they are denatured, the shape of the protein will be deformed. As a result, the activity will also start decreasing. It is same in case of pH also. You can see in the graph that with the increase in pH, the activity has increased. There comes a point where the activity is maximum, that is called optimum pH, and then again the activity, it decreases because of the denaturation of the protein. So at lower temperature and lower pH, they are inactive, and at higher temperature or higher pH, they are denatured. This finishes up your chapter. So kindly go through all the assignments and the links which were shared earlier with you. All the very best for the CT.